Meet George. George is 20 years old and is living a good life. Right, George? But within the next 10 to 20 years, he will start to experience symptoms such as involuntary movements, depression, and poor coordination. Sorry, George. Eventually, in his 30s or 40s, George will be diagnosed with Huntington's disease, a progressive brain disorder that causes uncontrolled movements, emotional problems, and loss of cognition. So what causes the disease? The affected gene that causes Huntington is IT15 and is found on the short arm of chromosome 4. This gene is expressed in the form of protein Huntington in all mammalian cells, but there are higher levels of gene expression in the brain. All IT15 alleles have a section of trinucleotide repeats of CAG, cytosine, adenine, and guanine. Usually the number of repeats vary, but there are always less than 36 repeats in the normal allele, while the mutant allele has more than 36. These repeats code for the amino acid glutamine. During protein synthesis, the gene is transcribed into mRNA and translated into amino acids, forming a polypeptide chain. The CAG repeats creates a polyglutamate section within the polypeptide. The polypeptide chain folds to form a normal Huntington protein. The protein is essential for proper cell division and apoptosis. The interaction with other proteins in the brain, including those involved in transcription and cell signaling, helps support neurons and brain function. The protein transcribed from the mutated allele has extra long polyglutamate chains. When these chains fold, they alter their shape, changing the function, damaging the brain. Since the gene is located on chromosome 4, it is an autosomal disease. Moreover, it has a dominant pattern, meaning that only one copy of the altered gene is enough to cause the disorder. Therefore, most people with the disease is heterozygous. Children of carrier parents who have a 50% chance of inheriting the mutant allele, so those who do inherit it will develop the disease when they become older. As the altered gene is passed from one generation to the next, the gene undergoes anticipation. The size of the repeat increases, resulting in people showing earlier signs and symptoms. The mutated protein targets the striatum, the part of basal ganglia. It serves as for planning, habit learning, and modulating movement. Eventually, other areas of the brain are attacked, no brain structure completely spared. Since glutamine is polar, an excess causes proteins to form protein aggregates. As a result, proteins lose their ability to carry out their proper function and become toxic to neurons, resulting in loss of brain tissue. This reduces the ability to walk, talk, reason, remember, and includes involuntary movements such as chorea and dystonia. Behavioral symptoms such as mood swings and depression occur as well, eventually declining to dementia. Currently, there is no cure, but we hope there is one in the future.